Assalamu alaikum warahmatullahi wabarakatuh. Welcome to my YouTube tech channel. My topic name: What is genetic engineering? Hope so. Today topic is started. Genetic engineering or recombinant DNA technology involves the artificial synthesis, modification, removal, addition, and repair of the genetic material. Genetic engineering developed in the mid 1970s when it became possible to cut DNA and to transfer particular pieces of DNA from one type of organisms into another. As a result, the characteristics of the host organisms could be changed. If host organisms is a microorganism such as a bacterium, the transferred DNA is multiplied many times as the organisms multiplies. Consequently, it is possible to obtain millions of copies of a specific DNA inside a bacterial cell. Objectives of genetic engineering. The important objectives of engineering are as follows: isolation of a particular gene or part of a gene for various purposes, such as gene therapy, production of particular RNA and protein molecules, improvement in the production of enzymes, drugs, and commercially important organic chemicals, production of varieties of plants having particular desirable characteristics, treatment of genetic defects in higher organisms. Basic steps in genetic engineering. All the above mentioned objectives can be obtained by some basic methodologies such as isolation of the gene of interest. In the first step, the genetic engineer identifies the gene of interest in a donor organisms. Special enzymes called restriction endonuclease are used to cut the identified gene from the total DNA of donor organisms. A vector is selected for a transfer of the isolated gene of interest of the host cell. The vector may be a plasmid, the extra chromosomal DNA present in many bacteria, or a bacteriophage. The gene of interest is attached with the vector DNA by using endonuclease breaking enzyme and ligase joining enzymes. The vector DNA and the attached gene of interest are collectively called recombinant DNA. Recombinant DNA is transferred to the target host. In this way, host organisms is transformed into a genetically modified organisms. The GMO are provided suitable culture medium for growth to give as much copies of the gene of interest as needed. Before genetic engineer 5000 shape brains were required to produce 5 mg human growth hormone. The GMO contains the gene of interest and manufactures the desired product, which is isolated from cultured medium. Hope so your topic is finished. Inshallah, next topic coming soon. This channel, like, subscribe, बेल आइकन लाजमन। इसी से इजाज़त पाऊँगा और आपको इसी से ही कहूँगा। अस्सलाम वालेकुम रहमतुल्लाहि व बरकातहू। अल्लाह हाफिज़।